What happens if you want to modify? You want to induce a mutation. Up till now, the previous mini lecture, talking about, oh my gosh, these point mutations, these insertion deletions, bad thing, oh my gosh, these are natural occurrences, or you're exposed to some man-made chemical. But what if we want to go through and we want to make a mutation occur? We want to make a mutation under our terms, under our parameters. We refer to this as gene editing. You want to change the sequence of a gene. You want to insert a nucleotide sequence. You want to remove a nucleotide sequence. That way the gene, whether it's something's been inserted or deleted, that gene will now be the blueprint for an altered polypeptide. Gene editing is thought to be the future of genetically modified organisms. The plants that we grow, we harvest, the animals we eat. It's also thought to be the future of modern medicine. Why sit back and wait to see if, how bad mutation in somebody is going to be? Something silent, something deadly? Is it going to be cancer causing? When we can go in with an enzyme and then we can modify at the molecular level the DNA of the patient. You've probably heard of CRISPR, Cas9. Cas9 is a protein, an enzyme, or more specifically, a ribozyme, I guess it would be. It's components of protein and RNA. Not sure if it is an enzyme or a ribozyme. We'll just go with enzyme for now. Okay, it's a protein. Uses RNA to guide it to specific sequences of viral DNA, viral RNA, to go in and cut. Cas9 is a defense protein, a defense mechanism used by prokaryotes to genetically modify, genetically damage viral genomes. Damage the viral genome, cell's not going to die, cell's not going to be affected. Cas9 will cut at specific nucleotide sequences. Those nucleotide sequences that it'll cut are determined by small RNA fragments held by the protein Cas9. These RNA fragments are used as guides to look for the complementary sequence within the viral genome. Go find that sequence, cut. Find the sequence, cut. Well, guess what? We've figured out how to alter the RNA guide. This works great if you're discussing a prophage, a virus that has inserted into the bacterial genome. Okay? The viral genome is now hiding in the circular bacterial genome. So Cas9 will go along until it finds the DNA sequence that complements the RNA fragment that it's using as a guide, and it will cut, cut, cut. And then it will splice the DNA together. So in essence, the viral genome that is inserted into the bacterial genome will be cut out. Yeah, we figured it out. We humans have figured out how to modify Cas9. Modify Cas9, so not only does it cut out and remove certain sequences, but it'll cut and it'll allow for the insertion of a specific sequence, a specific DNA fragment. So up till now, we have been trying to figure out a way to deliver DNA fragments that would have the blueprint for the correct nucleotide sequence to yield a correct primary structure for a polypeptide. We haven't been able to find a good delivery mechanism. Try viruses. Well, viruses aren't always going to be able to go to the right cells. They're not going to be able to go and insert correctly. Then you have to worry about the patient's immune system clearing it out too quickly or killing the virus-infected cells. Okay, then we've tried just injecting directly DNA. Some of that DNA, will get taken up by cells, but you run the risk of the immune system recognizing that DNA as something foreign, and then next thing you know, you're mounting an immune response to all DNA in the body, which would be bad. CRISPR shows a lot of promise. Now, we've already been using it to modify, plant cells, to modify bacterial cells, to modify animal cells in culture. I do believe they've done it in a few mouse models. So we're waiting to see what can we do with humans. Now, side note, there have been a few human studies done that were illegal. Genetic modification using CRISPR of developing fetuses. Don't care what you think. That is horrendous. That's Nazism, Nazi-ish level human studies. I forget the name of the Chinese researcher who did that. He claims he destroyed the embryos after a certain certain stages of fetal development were met. He just wanted to show that it could be done in a developing fetus. Don't know if that's true or not, if there's been any more human testing, but yeah, we'll wait and see on that. But think about it. If we could show that CRISPR is not Frankenstein's monster, we're not going to be randomly mutating, randomly destroying genes, that even though you put the Cas9 protein in, with a specific RNA fragment, that it's not going to inadvertently swap that out for another chunk of messenger RNA that you have in your body, in your cells, and then indiscriminately start cutting. Think of the different diseases. Sickle cell, previous lecture. The previous mini lecture I was talking about the different point mutations. Sickle cell could be a thing of the past. You could take a child, an adolescent, an adult, treat them with, a CRISPR that has that sequence, fragment in it, so that it goes, it cuts, it repairs, inserts the correct DNA. Boom. From that point on, the red blood cells will be turned over in time, but from that point on, any new red blood cells made will have a correctly functioning hemoglobin molecule. But we got to be careful. Frankenstein's monster hangs over our head. All good intentions say, yes, this would work, this would be great. The problem is that we found with many, many things, what works great on paper, what works great in a lab, in tissue culture, what works great in a mouse model, can end up really messing humans up because we're not mice. We're not single cells growing in a petri dish. We're very complex organisms. And we humans are very, very brittle. We break, we get messed up very easy compared to our rodent mammal brethren.